to talk about what is cancer, what is the incidence of cancer in the world and also in India, and the genetic basis of origin of cancer. So it is a very, very important disease worldwide. Probably it is the second most common cause of death worldwide, the first being coronary artery disease or इधर बुला लो, इधर इस तरफ, ठीक है, आने दो सबको. So what is a cancer? Basically it's a collection of many different disorders, and it's not uniformly categorized by even the tissue of origin. So it is cannot confine to one particular tissue. The example being hormone receptor positive breast cancers. They have different biology, they have different prognosis, and they have different treatment as compared to hormone negative breast cancers. So if the hormone positive breast cancer is there, on the other hand there is hormone negative breast cancer, they have different approaches. So tissue of origin is same, but the behavior is different. One important feature that is shared by all human cancers is that they have a genetic basis. So most of the cancers have a genetic basis. Familial cancer syndromes are relatively uncommon compared with sporadic cancers. So genetic mutation is more important than running of cancers in families. Both heritable and sporadic cancers require the accumulation of DNA mistake or alteration. So there are two problems. The DNA either has mistakenly changed or it has altered in its integration. This leads to a stepwise progression of events, ultimately causing a cancer. So this is how cancer development process goes on. This is a normal cell. Then, as I told you, genetic influences. Genetic influences would produce a morphology change in the cancer cells, those which are destined to become cancer cells. This is the doubling of the cancer cells, then quadruple, and then multiple times it goes on multiplication. So the defect is a halt in the arrest of growth of cancer cells, and this is the example of colonic cancer. The cancerous cell division leads to a malignant tumor. Now these are the genetic mechanisms. So direct DNA influence can be done by chemical carcinogen or radiation or certain viruses. So this is a normal cell, then this is the DNA damage. Sometimes the repair becomes successful, but when it is unsuccessful, it can lead to mutated cells and rapid multiplication of these cells known as cancer cells, and then it turns into a malignant tumor. So you have to have DNA alteration through chemical carcinogens, radiation, or particularly some of the viruses. Then there is an interaction of genes which affect apoptosis. What is apoptosis? Cell program death. Then activation of oncogenes. This is the activation and inactivation of anti-oncogenes, growth promoting genes and growth arresting genes. These are the apoptosis regulating genes. So they are disturbed, and this leads to clonal expansion and tumor progression, leading to a malignant tumor. Carcinoma is cancer of the skin and other tissues surrounding internal organs and glands. Leukemia is majorly a cancer of blood cells. Sarcoma is a cancer of joint, muscles, lymph vessels, and cartilage. Sarcoma is a cancer of joints, muscles, lymph vessels, and cartilage. You must remember these terms. Lymphoma is a cancer of the lymph node and lymphatic system. Now, what are the basic characteristics of cancer, the hallmarks of cancer? Cancer cells are self-sufficient in growth signals. They induce growth by themselves. They are insensitive to anti-growth signals. The arrest of growth process is not there, so it goes on proliferating. Evasion of apoptosis, that is programmed cell death. Limitless replicative potential. So it has the capacity to multiply and multiply manifolds. Sustained angiogenesis. So what is the significance of angiogenesis? वो लास्ट बेंच वालों को इधर ले आओ, लास्ट बेंच वालों सब इधर आ जाओ. What is the significance of angiogenesis? Because for survival of these cancer cells, you need nutrition, and nutrition is provided by angiogenesis, that is new vessel formation, which is important for survival. पीछे जाओ, पीछे जाओ, पीछे. Survival of the cancer cells. And another hallmark of cancer is that it leads to tissue invasion and distant spread, which is known as metastasis. Reprogramming energy metabolism, that is through vessels and through nutrients, the cells are sufficiently nourished in order to survive. Evading immune response, the body's immunity cannot contain the cancer cells, and these days that's why immunotherapy is being practiced. Genomic instability and mutation is another hallmark, and tumor promoting inflammation. So these are the basic hallmarks of development of human cancer cells. The WHO estimates that cancer is the first or second. In some countries it may be the first, in other countries it can be the second leading cause of death before age 70 years in the majority of countries. In 2020, the statistics mentions that 19.3 million new cases of cancers were diagnosed globally, and almost 10 million deaths were attributed to cancer. Among women, breast cancer is the most common, 24.5% of all cancers, followed by colorectal cancer. Breast cancer is the leading cause of cancer mortality, followed by lung cancer. Among men, lung cancer is the leading cause of incident, following by prostate cancer. And lung cancer is the leading cause of cancer mortality, followed by liver cancer. The mortality in liver cancer is 10.5%. The burden of cancer varies substantially by geographic region, is impacted by factors such as age of the population. Aging population is more prone for cancer, prevalence of risk factors in the society, screening and treatment. What screening is being done to detect cancer at an early stage, and what treatment is available in the country, in the community, for containment of cancer cells. Country's social and economic development is also important. The incidence and mortality of cervical cancer is quite low in high-income countries as compared to low-income countries because of cervical cancer screening and vaccination for HPV, human papilloma virus. So screening for cervical cancer is very important, and vaccination for of HPV, human papilloma virus, at a adolescent age group in females. The cancer registry is there, and in northeastern region, the highest incidence of cancer in India is observed. Cancer of the lung, mouth, stomach and esophagus were the most common cancers in men. Head and neck cancers, lung. Breast and cervix cancer, the most common cancers in women in Indian registry. Highest burden of cancer breast was observed in metropolitan cities. Leukemias and lymphomas were the most common type of childhood cancers. Mouth cancer incidence rate was high in population-based cancer registry in Western and Central India because of the practice of smoking and tobacco chewing. There is a rise in trend of incidence of cancer breast, while cancer cervix is on the decline because of the preventive measures. World's highest rates of cervical cancer, gallbladder cancer, oral and pharyngeal cancers are in India. And this is the chart. You can see the female breast. In females, it is very common. Lung, prostate, colorectal, stomach, liver and cervix. So these are the top cancers which are found globally. Female breast, lung, prostate, colorectal carcinoma, stomach and liver. Rest of these are rare, but they are still present. Again showing new cases of cancer worldwide. So new cases of cancer, lung is the most common, followed by breast, which is almost in equal incidence to lung, colorectal cancer, cervix cancer, prostate cancer, esophageal cancer, stomach and liver. So these are another top cancer in the world. This is again various areas showing the preponderance of cancer. As I told you, in males, prostate and lung cancer are the two most common. In female, it is the breast and the colorectal carcinomas. Now, what are the demographic risk factors, cancer risk factors? Increasing age is an important risk factor most cases. People aged 75 years or older are more prone to cancer. Age likely represents the accumulation of biological changes that influence carcinogenesis. There is DNA alteration as the age advances. These changes occur at different rates in different people depending on their underlying genetics and exposure across the life course. For example, in Japan, stomach cancer used to be a very common condition. For cancers that occur in both men and women, cancer incidences tend to be higher in men. Sex differences in cancers may be attributable to prevalence of relevant risk factors. Prevalence of smoking has changed over time, but current smoking and smoking related cancers continue to be higher in men in most of the countries as compared to women. Again, cancer deaths, one of the most important single attributing factor is tobacco consumption. Tobacco use and in particular cigarette smoking is a leading cause of death from cancer in the United States. Almost 30% of the cancer deaths are attributed to tobacco smoking. So what are the cancers which can be there with tobacco use or smoking? Lung, oral cavity, nasal cavity, nasopharynx, oropharynx, hypopharynx, larynx, esophagus, stomach. So whole of the airways, whole of the digestive system, esophagus, stomach, pancreas, liver, kidneys, ureter, bladder, cervix, as well as myeloid leukemia. So all these cancers are much more common when there is high incidence of smoking. Obesity and height. It has been linked with endometrial and esophageal cancers, and other cancers which are linked with obesity are kidney, liver, pancreas and stomach. So it is important that maintaining body health, body weight is important for prevention of cancers. Now, what are the basic biological mechanisms that can cause cancers when the patient has obesity or is of increasing height? Number of cells are much more in a patient who is taller than who is younger. So they are more prone for DNA alteration or genetic influences. So chronic inflammation, hyperinsulin, insulin resistance is important in cancer development. Insulin like growth factors, pathways, impact of obesity and circulating sex steroids. So you have to maintain a ideal body weight in order to prevent cancers. Adult height is positively associated with risk of colorectal cancer, ovarian cancer and breast cancer. Alcohol use is again associated with many cancers, and alcohol is responsible for about 4% of the cancers worldwide. So at the same time, you should also know what should be a preventive diet for cancer prevention. Eat a diet rich in whole grains, multigrain. Vegetable, fruits and beans are good for cancer prevention. Consumption of fast foods and other processed food, ultra processed foods, which are rich in fat, starches and sugars, promote cancer development. And consumption of red and processed meat again are important implicating factors. There is a dose-responsive relationship with increasing amount of alcohol conveying higher risk regardless of the type of alcohol consumed. So higher the dose of alcohol, the higher is the chance of cancer. Physical activity has been hypothesized to reduce the risk of cancer, and it is important that exercise, regular physical exercise should be done in order to prevent cancer. So physical inactivity and obesity would promote cancer development. So for cancer prevention, adults should get at least 2.5 hours per week of a moderate to intense physical activity or 1.25 hours per week of vigorous intensity physical activity, as compared to coronary artery disease, which is 150 minutes of brisk walking or moderate physical activity. So in cancer, this much is sufficient, but if 150 minutes, then if a patient does an exercise of 150 minutes per week, it is going to prevent cancer as well as coronary artery disease. Now we have talked of how the DNA alterations can occur, can be due to infectious agents, and what are the most important agents? H. pylori, Helicobacter pylori, implicated in gastric adenocarcinoma. So Japan had a highest incidence of stomach cancer, but preventive measures with the anti-H. pylori kit reduced the incidence of cancer in Japan. HPV, I have already talked, is responsible not only for cervical cancer but head and neck and anal carcinoma. So HPV, hepatitis B and hepatitis C for liver cancer. So they are the implicating viral agents which are responsible for cancer. The other viruses which are implicated are EB virus, which can lead to nasopharyngeal carcinoma, Hodgkin's lymphoma or Burkitt's lymphoma, HSP type 8 Kaposi's sarcoma, schistosomiasis, bladder carcinoma, and HTL virus, human T cell lymphotropic virus. It can cause T cell leukemia and lymphoma. So these are the basic etiological agents: hepatitis B, C, HPV, Helicobacter pylori, HTLV, etc. Helminth, schistosomiasis, etc. and lymphoma due to HPV and HIV. Now, hormonal differences. Several lines of evidence suggest that estrogens play a central role in the cause of female cancers. Risk of breast cancer increases with increasing circulating levels of estrogens. They are the hormone derivatives, they all are related to risk of cancers. Androgens have been implicated in the risk of breast cancer and prostatic cancer. In postmenopausal women, testosterone and other androgens have been positively associated with risk of breast cancer. And in men also, prostatic cancer and breast cancer may be due to androgen excess. Among women younger than 50 years, the risk of breast cancer is about 20% higher in women who are on current or recent use of hormonal contraceptives compared with non-users. So oral contraceptives are carcinogenic in patients who are younger or those who use oral contraceptives. And longer the duration, the more the chances of cancer. Radiation leads to DNA changes and is a very, very important cause of cancer. DNA typically cause mutations, copy number changes, gene rearrangements and chromosome copy number changes. So this is how ionizing radiation. So long-term effects of high level of ionizing radiation on the risk of cancer is best estimated from people who were exposed to atomic bombing in Japan in 1945 and large nuclear disasters in Chernobyl in Russia and Fukushima. So nuclear explosions, atomic bomb use and accidental leakage of nuclear material led to a high incidence of cancers in the community. The cancers which were seen in Hiroshima and Nagasaki were leukemias, and they were observed 3 years after exposure. And excess risk of solid tumor were observed 10 years. So leukemia was the first to appear. Later on, solid organ cancers also appeared after the atomic explosion. Sites of excess risk include bladder carcinomas, breast, lung, brain, thyroid, colon, esophagus, ovary, stomach and liver. So all these cancers are attributable to ionizing radiation as well. Now, coming to the genetics of cancer. Basically, it is a disease of cumulative DNA alterations, as I showed you in my first slide. And there is an imbalance of cell proliferation when compared to cell death or quiescence. So, if this ratio is disturbed, this leads to cancer generation or to decrease cell death or stasis. So, when this happens, the initial steps of carcinogenesis have begun. Genes that mediate cell proliferation, cell death are often the early drivers of cancer. Many of these genes are mutated or altered in diverse types of cancers. This is again the same diagram which I showed you. So, chemical carcinogens, radiations, viruses I have already named, genes affecting apoptosis, activation of oncogenes and inactivation of anti-oncogenes. Now, one of the important classical example of a genetic cancer is a chromosomal dislocation. There is a translocation of chromosome 9 to chromosome 22, which is known as, what is it known as? Excuse me, उनको लड़कियों को उठाना जरा पीछे इनको. Stand up. Yeah, हाँ ये वाली green shirt के पास वाली. इधर इधर ग्रीन शर्ट पे पहनी है ना, ये इसके पास हाँ, इसके पास वाली हाँ, खड़े हो जाओ. कीप स्टैंडिंग. ओके. सो व्हाट इज फिलाडेल्फिया क्रोमोसोम? So it is a translocation of small part of chromosome 9 to small part of the chromosome 22. This is what is known as Philadelphia chromosome. What happens if this fusion occurs? This results in the fusion of BCR and ABL gene, causes CML, which is a chronic myeloid leukemia. Okay, CML. So Philadelphia chromosome is the translocation between the arms of chromosome 22 and 9, leading to unlimited proliferation of myeloid cells. Now there are two important genes. One is the oncogenes and the other is the tumor suppressor genes. Oncogenes are accelerators of cancer growth. They drive abnormal cell proliferation through various mechanisms. Common oncogenes that are often mutated or altered in human include CRAS, BRAF, etc. So these are the names of the genes. Functions of oncogenes are diverse, but many mutations and alterations that occur in oncogenes constitutively activate critical growth promoting pathways and drive cellular proliferation. So two important genes, one is the oncogenes which promote growth, and tumor suppressor genes which suppresses the growth, but then there is a failure of this. Tumor suppressor genes acts as brakes of the cell growth. The normal function is to control cell cycling by monitoring and stopping proliferation when appropriate. Any disruption in the function of tumor suppressor gene would lead to a net gain in cell numbers. Tumor suppressor genes commonly in human cancers include TP53, CDK, etc. Most cancers have genetic alterations involving both tumor suppressor genes and oncogenes. So this is very important that you know about the tumor suppressor genes and the tumor promoter oncogenes. Now this is the examples of oncogenes. This is the figure of A, this is the maternal and this is the paternal. So any mutation of oncogenes act as the accelerator of cancer cell driving forward even when the cell should stop. So this mutation leads to acceleration of the small growth. The signal stop is not followed. Again, there can be a change in the paternal allele. So this again would lead to failure to stop the cell growth. Similarly, tumor suppressor genes are the brakes of cell cycle. So these are the bad job. Tumor suppressor genes are the brakes. If a mutation or alteration disables only the front brakes or only the rear brake, the M or the P, they are disabled, the cell continue to go forward through normal stopping signal. So tumor suppressor genes, they are the brakes, and oncogenes are the accelerators of cancer growth. So human body has trillions of cells, and each cell generally contains 2 copies of the genome. Mature red blood cell and platelet are among the few cell types that are devoid of DNA. And gametes, ova and sperm, have only 1 copy of the genome, thereby allowing the zygote to inherit a haploid gene from each parent. DNA obtained from normal cell is often referred to as germline because it is derived from parental ova and sperm, which are also called germ cells. So there are germ cell tumors also. So mutations of these DNA can lead to cancer development, and this is as I've told you in the initial phase, altered DNA methylation and other genetic alteration can lead to colonic epithelium from initial stage of adenoma to carcinoma and then to distant metastasis. Now there are certain syndromes, familial, these are not very common, but they are seen: Cowden syndrome, familial adenomatosis polyposis, APC. So these are the various familial cancers which run through genes. What are the basic testing for cancer genetic testing? PCR is one important, and FISH, fluorescent in situ hybridization, karyotyping, DNA microarray, DNA sequencing, and real-time PCR and methylation assays. This you should remember. So mutations can be found out by these methods like FISH for Philadelphia chromosome, etc. They can be identified through these types of genetic testings. So this is in summary: PCR, real-time PCR, karyotyping, FISH, microarray, DNA sequencing for genetic testing, which is easily available these days and should be done. Now, when you have identified the genetic information about a cancer, you can use this to assess the risk, what is the prognosis, what is the diagnosis, what is required for screening, and what is the therapy which you should give in these patients. And these are known as targeted therapies. They are based on an understanding of how oncogenes and tumor suppressor genes drive cancer growth and develop strategies to disrupt these mechanisms. So, breast cancer, etc., they are dependent on receptor positivity and the genetic testing. So, you can use targeted gene therapy, targeted therapies directed against these cancer cells. Now, finally, coming to multiple choice questions, commonest cause of cancer is? Common cause of cancer and prevalence of it is tobacco 22%, infections 15%, lifestyle changes 10%, and genetic defects 10%. On the background of genetic mutations. Factors that cause cancer: tobacco, chewing, smoking, excess alcohol, obesity. So this is the summary. Every single agent can be responsible for cancer development. Tumor suppressor genes. What are the statements which are true? Except tumor TSG are class of genes that suppress the activity of tumor development, natural protector of our cells, acquired germline mutation, the tumor suppressor gene produces loss of function, mutation in the class of BCR-ABL gene can increase the risk of hereditary breast cancer. So, answer is D. This leads to CML, chronic myeloid leukemia. Genetic testing for cancer may help for detection or presence or absence of cancer, staging of cancer, identification, detection of chance of inheritance of cancer. So, not only these factors, but you can also know the chance of inheritance. So, all of the above: detection or presence of absence of cancer, staging of cancer, identification of genes, detection of chance of inheritance, prognosis of a cancer, monitoring the recovery of cancer. Finally, a word about prevention. Avoid smoking and chewing tobacco, less alcohol consumption, maintain a healthy diet by eating lots of green vegetables, fruits, antioxidants, rich foods and grains. Stay away from excessive sunlight, use sun creams. So, this is how the cancer control continuum goes on. Find out the etiology, prevention of lifestyle abnormalities, early detection, diagnosis and treatment, and then end of life care. So, this is all about cancer, common malignancies and the genetic inheritance.